drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Smith, thank you for joining me.
Hi.
Do you want to start off by just telling everyone a little bit about who you are and what you study?
Yes, I'm a medical epidemiologist. I'm director of the Medical Research Council Integrative Epidemiology Unit at the University of Bristol. I'm particularly interested in attempting to get as strong causal evidence as one can from all different types of data, and I've worked across many different substantive fields over 40 years, but always with a focus on causation.
So for those that don't know formally, what is the field of epidemiology? What is that field concerned with, and what are its major tools?
So epidemiology studies influences on disease, and its tools include all forms of observational studies, from sort of ecological studies. If you see that disease rates are much higher in some particular areas than others, that gives you a clue to the cause. And then formal studies that are set up to so either to hypothesize, test, or to study a large number of factors in a more hypothesis-free way. Studies could be case control studies, where you take people who've had a disease and then you try to get controls, people who haven't had the disease but who are similar to the patients the people who have developed the disease. And then you look back retrospectively to see the factors that might have influenced them getting disease. I mean, like everything I say here, these are informal definitions. There are formal definitions of how you would sample in case control studies, but I'm going to obviously discuss the sort of informal definitions. So, and then there would be prospective studies when you recruit a group of people within a population. These can be, based like within industries, famous studies of civil servants in the UK, famous studies of nurses and male health professionals in the US. And you gather data on those people when you recruit them into the study, and then you follow them forward until they develop a disease. And then you look at the factors then that predict them having developed disease. And then there are trials when you randomly assign people to interventions, or you can randomly assign groups in cluster randomized trials, or indeed communities, as has been done. And then you randomize an intervention that will modify the factors that you think are important and see what the outcomes are.
And so you've got epidemiology, you've got randomized controlled trials. There's this concept of the hierarchy of evidence in medical science and human health. Can you talk about that a little bit? What is this hierarchy of evidence? What are the major types of studies beyond controlled trials and epidemiological studies? And how do you think about that hierarchy and its structure, and like what it means in terms of something being at the high end or the low end of it?
Yeah, so I don't like the notion of hierarchies of evidence, because you often see these pyramids drawn. It'll have randomized controlled trials at the top of it. Then we'll have like prospective observational studies, or sometimes these days you sometimes see Mendelian randomization put as second, which we might get onto, and then it goes down. All studies can be bad. I mean, randomized trials, they can be just faults that occur in instigating the studies, in carrying out the studies, and there have been, well-known cases of this. So you wouldn't put some badly done randomized controlled trial above a well-done prospective observational study. So it's a useless notion that should be past its sell-by date by now. I think one's got to think of ways of actually putting together the information that comes from different study types, and thinking of ways of combining the data that can be combined, thinking which data you actually think deserves combining, and coming to conclusions in that way. Ways which a sort of approach that has been referred to as triangulation of evidence. And that is very much runs against the hierarchy of evidence sort of notion.
Yeah, and I want to get into some of that throughout our discussion today, how you think about, good ways or good strategies for triangulating evidence across different study types. But talking a little bit more to start on randomized controlled trials, obviously they can be very informative when they're done well. But oftentimes what you see out there in the zeitgeist is people are thinking in terms of this hierarchy of evidence, and you often see terms like gold standard, RCTs thrown out there. And oftentimes, not always, but oftentimes people just sort of regurgitate the headline result of a randomized controlled trial, assuming that it's, it's branded gold standard, therefore it must be good. I want to give people a clear sense for what the strengths and weaknesses of these studies are, and the types of ways they can be designed poorly or well. So what are some of the common limitations in practice and the common assumptions that go into randomized controlled trials? I'm thinking of things like, blinding, adherence to treatment, attrition from the study, randomization, and so forth.
So as the name implies, the power in randomized controlled trials comes through the process of randomization. This is a way that means if you have a large sample and that the randomization is done properly, the randomization can't be subverted, then that means that you will balance out factors which will be confounding factors in observational studies. And confounding factors will be something which causes your exposure and also causes your outcome. And a joke example often given is yellow fingers and lung cancer. If you went across a population, people who've got yellow fingers would have a higher risk of lung cancer than people who haven't, because yellow fingers will come from smoking. Obviously, scrubbing people's yellow fingers clean isn't going to reduce their risk of lung cancer. But the smoking causes both yellow fingers and causes lung cancer. That is a confounder. Probably the most problematic confounder for observational studies is reverse causation. And by reverse causation, you mean that you're talking about the disease process influencing both your exposure and influencing your outcome. Now, importantly, and how people have got this wrong, is that it can be the disease process before the outcome occurs. So, once someone's had a heart attack, or once someone's died even, obviously you can't get reverse causation from death to anything, because death, you're dead. It can't have influenced your exposure. But if you think of a disease like coronary heart disease, where atherosclerosis starts in relatively early life, soldiers in the Korean War, US soldiers in the Korean War, and autopsied at 19 already had a fair amount of atheroma. And atherosclerosis is an inflammatory process. And so that influences many things, including factors like C-reactive protein, which is an acute phase reactant. And but that's influencing it years, decades before you develop your heart attack. So you can have something which is a really good predictor, or is a good predictor of the outcome, measured decades before the outcome, but is caused by the disease process influencing the exposure. Now, that is virtually impossible to get rid of. And when you hear the term, statistically controlled for, then you sort of become suspicious, because you cannot statistically control for all possible factors, especially when you consider that they're going to be measured with error, they're going to change over time, there'll be ones you haven't got, etc. So it gives a false sense of security. Oh, we controlled for this, and this.
Yeah.
So now randomized controlled trials avoid that. That is the magic of randomization, and a really well done randomized controlled trial is strong evidence. Unequivocally strong evidence. I mean, yeah. So
yeah, I want to. Yeah, so you said you can have these false senses of security just because you statistically control for something doesn't actually mean necessarily that you know exactly what's going on or what the direction of causality is. And I think that's an important concept for people to understand, is the direction, like directionality in these cause and when we think about cause and effect. Does A cause B, or does B cause A? Oftentimes people go into studies with a very strong suspicion of the direction, and they can forget that things can go in a different direction, or that you can have a confounder, or that you might not be able to statistically control for it even if you have the data.
Yeah, no, I mean, absolutely. I mean, that is the problem of observational studies, especially if done naively, without introducing many of the ways you can actually strengthen evidence from observational studies.
Another thing I want to ask you about is if there's ever a false sense of security that comes from something like the sample size. So oftentimes people really lean into the sample size of a study, including a randomized control trial. And when it's done naively, in my view, they'll say, this is a very large study. It's a large N. It's a large sample size. It's a representative sample of the population. And therefore, it must be good, or the results must be very, very, very robust. And I want to get your take on that in the context of thinking about large studies where we're just looking at the average. And, what do you think about, so one way of thinking about this is, well, you want your sample size to be large because your statistical analysis will be, that much more compelling, perhaps. You want it to be large in the sense that you're including samples of humans in a randomized control trial, say, that are representative of the general population you're sampling from and trying to understand. But then you've also got the potential problem where if you're just looking at, say, the average of a large sample, and your sample is a bunch of different subpopulations which are systematically different from each other, there may be very few individuals who actually look like the average, and the average isn't actually, capturing what's true for any given subpopulation, say. How do you think about sample size and things like that?
Yeah. So there's lots of points, and I'll I'll go through them if I remember them. So in an observational study, that when the results may be confounded, then all that happens with the larger and larger sample sizes, you get tighter and tighter confidence intervals around an association where the exposure is not causal. So, if your sample size of people with yellow fingers gets to the size of UK Biobank, 500,000, you'll have a sort of 10 to the minus 1,000 p-value. Unequivocally, these are linked. And a problem with very large sample sizes or large sample sizes is that they give this notion that things are true because they are statistically robust. Indeed, there was a nice preprint out recently which actually looked at when information was presented to people about a series of hypothetical studies. When the sample sizes became larger, the people were more likely to interpret them as causal. Now, it is the case that you have sampling variation. So, and some things, are just chance. But once you get to very large sample sizes with reasonable numbers of events, that ceases to be a problem. And you can see the issue is that if you have large sample size through, for example, doing a systematic review and a meta-analysis, then if all of your studies come from populations where the same confounding is happening, then you get the same situation. It's spurious precision, and it translates into people thinking, thinking that the results are actually meaningful, meaningful in relation to the exposure and the outcome. I just want to clarify one thing is that you often hear people saying, correlation isn't causation, correlation doesn't imply causation, etc. That is wrong. If you have a robust, reliable correlation, there is causation in the data. It is simply that the causation isn't between what you might think of as the exposure and the outcome. So when you show yellow fingers predict lung cancer, that is there because smoking causes them both. You do not get reliable correlations that replicate across studies because of chance.
Right.
So reliable associations do imply causation. And one of the things which I find disappointing across the decades is how people have carried out observational studies that suggest things which turn out to be not true, don't go back and work out why they've got the wrong answer.
Right.
Famously, and it addresses a lot of these issues. Two papers appeared back to back in the New England Journal of Medicine showing that vitamin E supplement use was related to a considerably reduced rate of coronary heart disease, about 35% lower rate of coronary heart disease, and they controlled for everything. And they did two papers, one from the Nurses' Health Study, one from the Health Professionals Study, and they both got the same answer. So then you get this incredibly reliable looking association. Now in this case, the exposure they looked at was taking vitamin E supplements. That was the exposure. They asked people, are you taking supplements containing vitamin E? And if people were taking those supplements, they had a substantially lower rate, a risk of coronary heart disease. That was the exposure, and it replicated. Trials were then done of randomizing people to vitamin E supplements, precisely the exposure that was measured in the studies, and they were not. What happened was about half of the participants in those two studies started taking supplements containing vitamin E when the results came out. They were splashed across the front page of the New York Times, saying vitamin E greatly reduces the risk of coronary heart disease. making a causal claim in its language, and people aren't stupid. They started taking it, and when the trials came out, they stopped taking it. Okay, so the methodology, you think that there's something wrong with the methodology. Clearly, there's a very obvious confounders in that situation. The people who are taking vitamin E supplements are health conscious and have many other factors in their lives which are related to reduced risk of disease. But when the trials came out, no one went back to say, why did we get the wrong answer? What they went on to do was then publish papers about about folate and many other factors that appeared to related in the same way. The higher levels, lower risk of disease, or lower levels, higher risk of disease, or whatever. And then trials were done for those things. There's a list of about 12 things that came out where large-scale randomized controlled trials were carried out and showed no benefit, despite what appeared to be really reliable observational associations and well-done prospective studies. There must be something causal underlying that. So you should go back and try and work out why you've got the wrong answer. In fact, funding agencies should say go back and work out why you got the wrong answer last time.
Well, I want to talk about a little, like, why do things play out that way? Why didn't they go back and work things out as you're suggesting, but instead they sort of do another study and then they do more trials? Like, that's a lot of time, it's a lot of effort, that's a lot of resources that were effectively wasted because people didn't really go back and dig into the original observation. Why do things play out that way? How is that tied to, say, the currency of the profession? Because because even though people don't go work things out and it's sort of like this wasted effort when it's all said and done, all along the way, the people doing this are getting publications, and like there is a clear reason why they're doing things this way. The reward in academic systems is through publication. That leads to job positions, etc. It helps getting grants. If you show things are not the case, that doesn't lead to getting grants and academic position in the same way. So that's why it's that the reward system is sort of broken in academia. I want to say that this situation is not anything specific to epidemiology. Exactly the same thing will be happening in people who are doing, who are doing tissue culture studies, doing mouse studies, doing all forms of science suffers from this. That people don't go back to find out why things that were put forward were wrong, things that they put forward were wrong. It's nothing specific to epidemiology. I want to talk to you a little bit more about the interaction between, earlier we said, you mentioned the vitamin E studies and how, these things were reported on in the media. So then people hear about them and that affects their relationship to vitamin E supplementation. So in a very concrete sense, the science that gets published can get talked about in a public way and come to affect people's behavior. And these things all interact. When we think about industries like the supplement industry, which is very big, when we think about things like nutrition, which is, always top of mind for people, it gets a lot of attention. Journalists love to write about it. People love to talk about it and think about it. And, what supplement should I be taking? What diet should I be doing to lose weight? And so on and so forth. There's really entire fields built around this type of stuff. One of them that you've written about is nutrition epidemiology. I just want to get your general take on how this whole ecosystem sort of works and what we should be wary of. And I don't just mean like the field of nutrition epidemiology, but the whole social ecosystem of, the media will write about the results. It will get hype that will influence people's behavior. This will influence what does or doesn't get funded. Like what is sort of your bird's eye view of this whole field and whether or not it's in a healthy state from a rigorous scientific perspective?
I think often fields just talk to themselves. That's is one issue. I mean, when I say that, other science, or all science has the same perverse incentives, etc., because epidemiology is about human health and people are interested in human health, and because it's looking at trying to identify factors which influence health, and therefore the information that comes from those studies is instantly translatable into things you can do with your life. That's why it has the very high visibility, and with that high visibility should come responsibility. And to my mind, responsibility in science is revisiting why things went wrong. I mean, it's often sort of said, the Popperian sort of notion of falsifiability is all well and good, sounds great, but it isn't actually what people do. Setting up the test, which is the hardest test for your hypothesis, and the rewards go with discovering something new. I mean, famously, journals are very reluctant to publish null findings. So that's another reason why one doesn't go back to that. I think if these things were framed in a positive way, such as saying, well, working out why we got the wrong answer to this question will actually will point at us at maybe will point us at important causes that haven't been identified. It can be seen as a positive action.
Mm-hmm. Another thing I want to ask you about is, no matter what field we're talking about or what type of study we're talking about, we can use nutrition epidemiology as an example, but there's many other examples we can use. When evaluating a paper, obviously the average person out there is often not going to be evaluating a study. They're not necessarily equipped to do that because it's not in their education. They might not care to do that because not everyone who is out there wants to read a scientific study. They might simply hear about it from the media. But if you are one who reads these things, either because you have formal training or because you're just very interested, I want to talk a little bit about how you evaluate studies in the abstract. And one place I want to get your take on is when you're looking at sort of how the data was acquired or how it was measured, because at least in my education, when I learned how to read these things and evaluate them, a kind of almost hack, I guess, that you eventually learn, or at least I eventually learned, is it's really easy to get focus your attention on fancy analysis and statistics and all of the stuff that is sort of put out front. But you have to actually go to the methods and look at how the data was collected, because more often than not, the data might not even be measured in a compelling way. And at that point, if you can't trust the input data, then it's sort of garbage in, garbage out, as they say. And so what is your general take on, evaluating the input data and how reliable that is across some of these fields we've been talking about?
So with the input data, what I look for is obviously, how they've collected both exposure data and confounder data. What you consistently see is a completely asymmetrical view of the exposure of interest and the confounders. And so, if a study is interested in a particular nutritional factor or biomarkers, circulating biomarkers, these things aren't restricted to nutritional epidemiology. Or physical activity or whatever, people will focus on getting better measurements of the exposure they're interested in. And I mean, the physical activity field is one where that has got way better. With people with wearables, with being able to get actually objective measures of reasonably long-term physical activity measures, you start categorizing the physical activity better. You also, get some notion about the reliability of the measures, the degree to which even in the best you can do, what levels of measurement and precision there are. And you'll quite often see people correct for measurement error, which you can do. It's quite very simple. A very simple thing to do is to say, well, we know that two measures taken at 2 times for this exposure, they relate in this way. So we just sort of multiply up the effect to take into account that we're diluting the association. So it's sometimes called regression dilution bias. Why a single measurement will not give you the effect that you see with better measurements, better characterization, when there's less error in the measure for actually reflecting long-term exposure. But then you don't see the same care being taken with the confounders. The confounders are then just banged in as they are, and then this is something that very often if you actually apply the same corrections to the confounders, that you will then see that the strengthened effect attenuates again often to the null. So what I look for is symmetricality. Is that it's often said that the real danger for a scientist is to fall in love with their hypothesis, and therefore when you're in love with something, then you spend more time focusing on it, thinking about it, and then you take everything else just as presented. So it's the symmetricality and performing analyses which give equal weight to these things, that take seriously getting the best estimate for the combined factors.
How do you think about the relationship between preclinical literature, animal studies, things like that, and randomized controlled trials and the human literature? So in the context of caring about human health and ultimately wanting to get to an answer for A causes B in human health, and thinking about ultimately the clinic, what's the relationship between the preclinical side of things and the clinical research? Perhaps one way to frame this is, when there are discrepancies between what we start seeing in some of the human studies and what we expect to see from our understanding of mechanism and biology from the preclinical side, how do we start to triangulate the truth from the clinical and the preclinical side? And you see all sorts of stuff here out in the zeitgeist. Some people that are interested in clinical research kind of completely ignore the preclinical side because it's preclinical. Some people extrapolate automatically from the preclinical side, prematurely. How do you think about triangulating things from both the clinical and the preclinical literature?
Yeah, so I think if you're actually going to think about actually trialing things in humans, you have to sort of move into human populations, and then you have to move into large enough studies to actually get findings about both on-target effects, the sort of beneficial effects you want to get, but also to adequately monitor whether there are adverse consequences. So again, that's the symmetricality principle. That you need to be symmetrical about benefits and risks, and spend as much effort monitoring them both. I think this is something that where the methods haven't been well developed for saying how do we actually combine evidence from human studies and from cellular systems, animal studies, etc. Because you can't use a sort of animal study or cellular system to say, if I change this factor by so much in people, I'll get this much benefit. There's no way that those forms of preclinical data can give you those sorts of estimates, which is ultimately what you want. But at the same time, it's very obvious. It's clear that those data will be pointers towards what you're likely to see, and should be included certainly in an assessment of things that you consider taking forward. I mean, there are some examples of just purely empiric things that have been observed, then taken forward from empirical data and turn out to be the case. Famously, there's sort of effects of Viagra were a side effect of an what was thought to be a possible antihypertensive drug. I think it was, and then they were then trialed for what they became used for. But particularly currently in the current situation, most drug target identification for drug therapeutics would rely on preclinical data, including genetic evidence, etc. And when putting everything together to try to evaluate causality, I think all those levels of data need to be considered. But the problem is, in much of the literature, what you see is you see biological plausibility is put in. So you have a paper which let's say just substance A or whatever it is, something has some beneficial effect on evidence of beneficial effect on coronary heart disease, and then you get some observational study, say, which supports that. And then in the discussion, you'll see people will say, animal studies have shown that substance A does this, or substance A squirted onto cells does this, or what, it'll be put in. But it's cherry picked. Mm-hmm. These days with Google, AI, etc., it takes about 30 seconds to come up with some you could equally find references that say, substance A in animal studies puts up blood pressure and substance A puts down blood pressure. And they're cherry picked and reported as this sort of biological plausibility, which is actually worthless. So so one needs to have a systematic appraisal of those sorts of evidence, and that is something that there's attempts at developing systematic reviews of animal studies, for example, to actually try and avoid that sort of cherry picking. But it's still very much the mode that you just find something which supports what you want to say. As I say, it's the falling in love with the hypothesis thing. It's not none of this is like motivated badness.
Right.
It's what sort of anyone would do unless conscious of the temptation.
Right. Yeah, you have to be conscious of the temptation. You have to be extremely careful, and that takes a lot of work. And it, whether you're just a consumer of the science and you want to know, like, what should I do to help my blood pressure, or you're someone doing the science, when you think about, just the practicalities of life and the incentive structures at play, at some point you've got to publish a paper and do something. At some point you've got to make a life decision about, X, Y, or Z, do this diet or that diet. And so you have to do a lot of extra work above and beyond what your life absolutely requires to actually be this careful.
Yeah. Yes. No. I mean that's right, and I think there's roles for journals in terms of what they should require. So to go back to some of these nutritional examples. There's a sort of series of approaches which can be used in observational studies, which provide sort of sanity checks as to what they find. One of these approaches is now called negative controls. It's an approach which has actually been used 100 years in epidemiology, but it's one of those things that when it was given a name, which is a memorable name, it's probably now better recognised. But this would be a situation where you would say, well, let's take something which is just not plausible could be caused by this exposure, and carry out precisely the same analysis as we do for the outcome that we've fallen in love with, the association we love. Let's do precisely the same for an implausible association. So back in 1992, there was a lot of interest and publicity about that smoking was leading to suicide. Cigarette smoking is very, very bad. But observationally, it was shown that there was quite strong association that greater amounts of smoking, heavier smoking, smoking and not smoking, related to suicide risk, about sort of
we're talking about tobacco smoking.
Tobacco smoking, yeah, threefold risk. And then again, the cherry-picked biological plausibility. I mean, smoking does so many things.
Right.
The probability that you couldn't find something which is in smoking, which you could have a plausible story about suicide, is zero. Well, nothing's ever zero, but you know what I mean.
Yeah.
Diminishingly small. And serotonin was hit on. And it was said, smoking is going to deplete your brain serotonin. I mean, the actual data for that at the time were fantastic. It's going to deplete your serotonin, and it's going to lead to suicide. Now, standing back as a sort of population scientist, you think, well, firstly, there's obviously socioeconomic factors, health protective behaviours, etc., which will all confound the association. There's also a lot of possibilities for reverse causation. People who are depressed, sort of self-medicating with smoking, or if you're depressed, you find it very, very difficult to give up smoking. So there could be the reverse causation from the sort of process that leads towards suicide, influencing your smoking behaviour. That seemed highly plausible to us. Anyway, we published a paper which actually had the title, I can't remember it exactly, but the title was something like "Smoking and Suicide: Demonstration of an Artifact in Observational Epidemiology." And now this was a rare case where a paper which was actually arguing against something being causal was published in a high-impact journal, The Lancet. The Lancet published our paper, because what we did is we had a huge study from the US, 1/3 men in the US, and you thought, what outcome can you not really come up with a biologically plausible reason that has some biological effect that it causes the outcome? And being in the US, we could happily demonstrate, well, not happily, but we could demonstrate that smoking showed just as strong an association, just as large an association with being murdered as it did with suicide. Now, thinking of a causal reason for that, the only one would be if the paramilitary wing of the Health Promotion Agency had gone out and started shooting smokers as the ultimate deterrent. But otherwise, it's rather difficult to come up with a plausible story. So that's what we showed, and that was an example of what's now called negative controls, and we did quite a lot of studies using that approach. I mean, despite the title, Demonstration of an Artifact in Observational Studies, and that paper has been cited. Our paper has been cited amongst the literature showing that smoking causes suicide. I mean, so it shows how difficult mindsets are. But these same the studies that produced these findings on the vitamin E supplement use and whatever, they'll publish it with the end point of interest to them. But if you published it with other endpoints like that accidental and violent deaths, etc., and you showed the similar associations, that would make that would cast serious doubt on the legitimacy of the association. And an epidemiologist called Dana Petiti did a beautiful study when work was being done on hormone replacement therapy, when it was thought that hormone replacement therapy reduced the risk of coronary heart disease. Lots of observational studies showed that, but there was lots of reasons to think confounding could generate such associations. And she looked at dying in car crashes, and showed that accidental and violent deaths, and showed that sure enough, hormone replacement therapy use was associated with as large a lower rate for accidental and violent deaths as it was for coronary heart disease. And then when the randomized trials were done, sure enough, hormone replacement therapy did not reduce coronary heart disease risk.
Yeah, no, I like that. The negative control, just showing whether the same exact analysis is going to give the same
Yeah,
seemingly positive result
yeah,
for something that's just totally implausible.
yeah, yeah, exactly. And you can have negative control exposures as well.
Mm.
It isn't just a negative control outcome. I mean, situations where we've used these a lot are in looking at intrauterine influences on offspring outcomes, because you have a pretty good negative control if you're looking at, say, maternal smoking, and does what effects does maternal smoking have on offspring outcomes. Then, if you think of most of the confounders you can think of, will be sort of family level confounders, and they will, and the father's smoking will relate as strongly to the confounding. And so, if you look at a situation where it is clear that maternal mother smoking during pregnancy does lower birth weight, and then if you look at the father smoking during the pregnancy, that isn't associated with the birth weight. Once you take the fact that smokers marry smokers into account, once that's taken into account, it doesn't relate to the birth weight of the offspring. But then, if you look at other outcomes, like whether the offspring gets a diagnosis of ADHD, okay, you find that the maternal smoking does relate to that, but so does the dad smoking. Relates in the same way as the mum smoking. So that doesn't look like an intrauterine effect. So that's a negative control exposure, where you have an exposure that would have the same confounding structure as does. You look at a negative control exposure, which has the same confounding structure as does actually your exposure of interest. Now, these are hugely underutilized in epidemiology. I mean, definitely their utilization is increasing. But journals could say, well, we're not going to consider your paper,
Right.
Unless you actually include some sanity checks.
Right. Because there's a natural incentive not to do these types of negative controls for the researcher, because every time you do it, it's one more chance for the exciting result not to turn out to be true. But at the same time, so the journals could gatekeep there. They could, or I should say, they should do quality control there. They should require these things. You could make that argument. It's obvious why, but at the same time, the journals also have this perverse incentive where they also want.
They don't want negative papers. No papers. No, exactly.
So how do you think about? I think there's lots of cases where there might be a human health related question of interest, and there's actually a lot of literature out there on it, but maybe there's never one definitive study that answers it, once and for all, or in all cases, or for all people. But we sort of have to hedge our bets and maybe come to a conclusion, at least when it comes to making, the practical decisions in our lives. So, maybe there's some randomized control trials out there. Maybe there's a lot of observational work out there on some health related question. I'll let you pick if there's a good example of this. I don't know if it's the relationship between, say, alcohol and health, where you see people argue it both ways. Maybe a little bit's good for you, none of it's good for you. You see learned people who know the literature argue things both ways. It's very common to see that. In a situation like that, how do you approach, sort of what stance to take, whether something's, potentially good or not good, when the evidence is out, there's evidence out there, but it's imperfect. There's this patchwork and you've got to triangulate something from it.
Yeah. So, I mean, alcohol is an interesting case because there haven't been randomized trials of alcohol use and its outcomes and its effects on outcomes. I mean, famously, with alcohol, the argument has usually been that alcohol reduces or lowers the risk of ischemic heart disease. That's the story which is most often told.
And that story is typically something like, 1 or 2 glasses of wine every day or two.
Yeah. Now, I mean, the interesting thing actually is, and this is a story, if people are actually interested in getting into actually how science works, it's a fantastic story, I think, is in fact the NIH did set up, or funded, should we say, they funded a trial of alcohol consumption, basically taking people who were not. The idea of randomized people who don't drink much and increase it a bit. As I say, it was called the MAC2 study. I mean, you should, if you're as I say, if you're interested in these sort of questions, this is really something that's worth looking up. The alcohol industry were involved in co-funding it, the alcohol industry wanted the trial to be called Cheers. That was the name, but they were having influences on what was going to happen in the trial. So it's known that alcohol, the evidence is reasonable that alcohol increases the risk of breast cancer. So they were wanting women with family histories of breast cancer excluded, etc. And the email exchanges between the investigators and representatives of the alcohol industry were eventually made public in a 200-page report. This is a great read, honestly. It's available. It's easy to find. That Francis Collins, who was then head of the NIH, instigated into it when he got inklings of this. There's these sort of redacted emails where it's quite interesting. You can actually work out who they were to by the length of names, but never mind. We won't go on about that. But honestly, I really suggest anyone who wants to know about the actual mechanics of science look into that, and rightly pull the funding, because there was just not good behavior, should we say.
I see. So basically it was revealed that in these emails there was undue influence happening.
Yeah, absolutely. Yeah.
Okay.
And as I say, you don't have to. It's entertaining reading the report.
But just to give people.
Okay, but we'll get back to how would I. So that this is an area I did a fair amount of work in, because. So there's a sort of a variety of methods. I mean, one thing is that there's the sort of the sick quitter syndrome. So there's the notion that people will give up alcohol because they're told to by the doctor. I mean, if you go to your doctor for anything, they'll tell you to drink less. So maybe that'd be a part of it, is people reduce their alcohol intake, because they've got high blood pressure, etc., illnesses. So there if you then think that in that situation, if you have a sort of a highly representative general population study which contains quite a lot of people who are ill, you would get more of an adverse effect at the low end of alcohol, which will make it look as though drinking to a certain extent is beneficial. I'm waving my hands about now, but I realise now I've been showing these curves, and I realise now that's it. But anyway, so you take populations like a sort of a population of only working people, where there'd be less people who are ill, and then and sure enough, we did this in two matching populations, and amongst a population which would be a healthier population on average, there wasn't this evidence of benefit that was seen in general populations, and you see that for many things, including like low body mass index apparently being good for you. Anything you see these J or U-shaped curves, it's often that there'll be reverse cause and confounding influencing the lower end of the curve. But with alcohol, I mean, alcohol was a good story for Mendelian randomization. So maybe then that will give me I'll say something briefly then about Mendelian randomization. Mm-hmm. So Mendelian randomization is using germline genetic variants. So these are the genetic variants that you are born with, which are given to you at conception, and is a random draw of variants from your mum and dad, and then you get them. And that's what you get. Now, the ideal Mendelian randomization study, and now quite a few studies are the ideal form is when you've got, say, siblings, more than one offspring, and then it really is true randomization. If you look between the siblings,
right,
the genetic variants they have got are random. They're random draws from the same pool. So
right, you've got two parents and two siblings, say a brother and a sister. Each one might get one of the two variants in each parent. Right, it's all contained.
It's totally random. That does have some analogies to a randomized trial. What they're being randomized to. However, the genetic variants may have more than one influence. That's the problem with a major issue with Mendelian randomization is that genetic variants might have more than one influence.
Yeah. Can we actually back up for a second? What is sort of the purpose of Mendelian randomization studies? Why do people do these studies? What are they interested in?
So you do the studies because you utilize genetic variants that influence a phenotype, influence something that might be an exposure. Mm-hmm. So, a classic thing would be LDL cholesterol.
Right. So not only can LDL cholesterol vary as a function of your diet and lifestyle, but there's also genetic factors. And some people might be predisposed to higher LDL, and therefore we might hypothesize they're more, say, predisposed to heart disease.
Yeah, and that's what it isn't generally that there's just one variant which has a large effect. Even though there are a few of these variants, they're sort of they're relatively rare. I mean, you see these large effect things with familial hypercholesterolemia, but also there's lots of, you can put together a number of genetic variants which have smaller effects, to generate a sort of to generate differences in LDL cholesterol between the groups who have the different variants. Now, the analogy with a randomized controlled trial there, if you did have the siblings, for example, would be that you then analyze by the group. You group people by their genetic variants, and then you do an intention to treat analysis. You don't take into account what their actual level of LDL cholesterol is at all. You don't use that. You just use the genetic variants they've got. Just like if in randomized controlled trials, you asked me earlier, about subversion of randomized controlled trials. Those are the sort of situations where there is a randomized trial, but then they just look at the group whose cholesterol level fell or something, and when you do a randomized trial, your first analysis has to be what's called an intention to treat analysis, which is you just analyze the complete group who were given stuff A, and the complete group who were given the placebo. You look, and then the confounders will be balanced, and that's the same in Mendelian randomization. The confounders are balanced. So if you analyze by genotype or by the genetic variant groups, then you have an intention to treat analysis. Now, in the case of alcohol, in East Asian populations, China, Japan, Vietnam, etc., reasonable proportions, 20% or so, and higher in some populations, will carry a null variant. That means it's a non-functional variant in a gene called aldehyde dehydrogenase 2. Now, this genetic variant is part of the alcohol metabolism chain. So when you drink alcohol, a variant called alcohol dehydrogenase converts that to acetaldehyde, and in cartoon form, acetaldehyde gives you all the negative effects of alcohol. So it gives sort of palpitations, headache. It gives flushing. That's why this sort of variant is often called, Asian flush variant. So people who have one copy of this null variant drink less than people who have two copies. And the people who have two copies of the null, drink less than people who have two the normal, of the functional variant.
Right. The idea is, right, your body metabolizes alcohol into acetaldehyde, I think. This is going to give...
and then sorry, the aldehyde dehydrogenase 2 clears acetaldehyde.
Right. So the more of that you have, the faster you clear it, and the less apt you are to get your headache and feel hungover and so forth.
Exactly. Yeah. So if you have 2 of the null variant, then people don't drink.
Yeah, it would feel horrible. That's why they don't do it.
There's very low levels of drinking, and so if you then look across the three, there's 3 groups. People have got 2 normal copies. People have got 1 null copy and 1 normal, and people have got 2 null copies. And then if you look at blood pressure in men, what you see is that the men who have 2 null copies have lower blood pressure. And that lower blood pressure is because they drink less alcohol. But then you have beautiful sort of negative control type thing in this situation, because in the populations when the data were collected in the study we published in 2008, which got together a lot of studies that have been carried out in the 1990s, etc. 2000, not as Mendelian randomization studies, they were just sort of genetic association studies. Women in these populations didn't hardly drink. So that is beautiful, because if the genetic variant has an effect on blood pressure, which is not through alcohol, then you would see the same effect in women and men.
Right.
But what you see is the lower blood pressure in the men who carry two null variants, but you see no difference in blood pressure in the women. Now, it's a bit of a challenge to come up with an explanation of how that can be true.
Yeah.
If it isn't that alcohol is influencing blood pressure, and then there's lots of other that triangulates with other methods, and much to my disappointment, I actually enjoy drinking. Much to my disappointment, using this method then to look at coronary heart disease demonstrates that there is no protective effect at any level of alcohol consumption on overall cardiovascular disease. Certainly on stroke, it's just linearly adverse on stroke, and particularly so for hemorrhagic stroke. All cardiovascular disease together, it's adverse, and because these populations have rather low rates of ischemic heart disease, the result for ischemic heart disease is less precisely estimated, but is not precisely estimated. So that's very disappointing. That alcohol is just adverse at all levels.
Yeah. So that's an interesting case where you can do this Mendelian randomization, but you also have a population where, I guess for cultural reasons, the men and the women are drinking differently. So it gave you this situation where you could sort of see this, because you had not only the genetic differences between individuals of both sexes, but also this population level difference where the men tended to drink more and the women not so much. And so you could really triangulate from that well.
Yeah. And you can do the similar things with smoking. There's a genetic variant and a nicotine receptor that relates to smoking more heavily if you smoke. It doesn't relate to whether you ever take up smoking. It does relate to how heavily you smoke if you do smoke, and how difficult you find it to give up if you smoke. And that means that for that variant, you can look at how that variant relates to disease in people who have been never smokers and people who are ever smokers. And you see that amongst ever smokers, that variant the variant related to heavier smoking is related to lots of adverse health outcomes, and amongst never smokers, people who've never smoked, it's unrelated. So those are beautiful sort of situations when you have such interactions. I just have to to say that these are postulated, and these are incredibly well established interactions. Basically, the name of the substance is in the name of the gene, nicotine, and it's a nicotine receptor variant. Alcohol dehydrogenase can also be used for these studies and shows similar things, but it doesn't have as large effect on alcohol consumption as the acetaldehyde. But there's alcohol dehydrogenase, acetaldehyde, or aldehyde dehydrogenase, which gets rid of the acetaldehyde. These were genetic variants that were identified because of their modification of the exposure.
What are some of the common limitations and assumptions of Mendelian randomization studies where, studies come out, but, maybe we're not paying attention to the underlying assumptions and we think something is sort of true or related to this genotype when actually there's something else going on?
Yeah, no, I mean, there's a large number of caveats. I mean, the pleiotropy, the fact that the genetic variant might be different, might be having more than one influence. That's a particular problem.
So pleiotropy is the concept like one gene can do two or more things. It can do many things, and we're often implicitly assuming that sort of one gene is involved in one phenotype, but nothing else.
Yeah, exactly. Yeah. So pleiotropy is a problem, and a particular problem is as these genome the studies which sort of discover these variants, and from many traits like body mass index, there's sort of 1,000 variants that statistically are robustly related to it, and the same is true for C-reactive protein, which I mentioned earlier, and many other things. The problem is that the larger and larger these studies go, the more likely it is that they're going to pick up, they're going to be picked up as a genetic variant for your trait of interest, but they're influencing your trait of interest through an upstream phenotype. So an example of that would be FTO, which was the first common genetic variant identified related to body mass index. Once you get huge GWASs, FTO relates to many other things because the genetic variant influences BMI, body mass index, and BMI influences many, many, many things, right, including C-reactive protein. Now, if you didn't realize that the genetic variant is working through a higher order, a higher level phenotype, upstream phenotype, and you took FTO to be a genetic variant for CRP, you're going to get completely the wrong answer because BMI does influence coronary heart disease. And therefore the answer you would get if you're using your genetic variant as a genetic variant for CRP, the answer that you will get will be completely wrong. So that situation is called correlated pleiotropy because the strength of the association of your genetic variant with your downstream trait or your genetic variants with the downstream trait will scale perfectly. So you will then get a whole set of variants which look as though they're giving the same answer. So you think, well, you become confident of it. And the issue with Mendelian randomization is many of the studies are just simply ludicrous because they're carrying out genome-wide association studies of things which it's just totally implausible. There would be a sort of direct variants that were not working through higher order factors like people GWAS skipping breakfast. I mean, sadly, this isn't a joke. There's a whole stack of papers saying, we prove that skipping breakfast is bad for you. Because, of course, genetic variants relating to many factors, educational attainment, body mass index, risk-taking behavior, all these things will relate to skipping breakfast, these upstream phenotypes. So, so Mendelian randomization can do things that Mendelian randomization can do. It does those really well. Mm-hmm. It's got the right answer on many things, which observational studies get the wrong answer on. I mean, famously, so-called good cholesterol, HDL cholesterol, which used to be called, even in popular parlance, good cholesterol.
Right.
Everyone knew that, and literally billions were spent on randomized trials raising HDL cholesterol.
Right.
Through different mechanisms, which had no benefit at all. did not
Right.
reduce.
The idea was, well, people still do this. LDL was supposed to be bad cholesterol. HDL was good cholesterol.
Yeah.
Therefore, it would be beneficial under this way of thinking to lower LDL cholesterol, which we do all the time with statins, say. Sort of symmetrically, it must be good then to raise the good cholesterol, HDL. You're saying they did those trials and there was no benefit.
No benefit. No. I mean, as I say, to a huge cost. And the Mendelian randomization studies came out about the same time as the first negative trial. But when the first negative trial came out, and this really is an example of people being in love with their hypothesis, it came out in the New England Journal of Medicine, and an editorial that went with it said, "Oh, this is it's because there's other bad effects of the drug. The drug raises blood pressure or something." So then they went on to do trials of something working through a completely different mechanism. That didn't work. And it took trial after trial after trial using different mechanisms. And in fact, and there's a beautiful sort of set of interconnected beliefs, which has been almost impossible to shift. That the one thing that alcohol does is it increases your HDL cholesterol. Randomized trials, you show you don't need massive trials to show that HDL absolutely, definitely puts up HDL cholesterol. So that was thought to be the mechanism. That, and then it also lowers fibrinogen, blood fibrinogen, which was thought to relate to blood clotting. And all these papers would appear, these pretty good journals, from very respectable universities, when they would say, we now show that 70% of the benefit of alcohol and coronary heart disease is through HDL cholesterol being raised and fibrinogen being lowered. And in fact, Mendelian randomization shows that fibrinogen isn't causal for coronary heart disease, and HDL cholesterol isn't causal for heart disease, but they were probably writing about that, I think, led me to my favorite line I've written in a newspaper, in a published paper, which was, I went through a lot of the work by this group, and then I said, there's one statement. I said there's one statement in their writing which is true, and the statement was that together HDL cholesterol and fibrinogen mediated 50% of the effects of the effect of alcohol on reducing coronary heart disease. And I said that is true because 50% of zero is zero. So that was a true statement that was made.
I want to ask you too about sort of when we're thinking about genetics and Mendelian randomization studies and this type of thing, when we're looking at, say, a biomarker that has a genetic influence, something like LDL cholesterol or whatever. Maybe you can give us a good example. My LDL might be at X because I've got some genetic influences tending it towards, level X. And someone else might be at an LDL level of X, but they don't have any of those genes that I do. They don't have any of those variants pushing it there. Their LDL's at that level purely due to, their diet, their lifestyle, whatever. Yeah. When we think about these genetics studies, when someone has, say, a biomarker like that's at a certain level, in part due to genetics, and other people have the similar level of that same biomarker, but not due to genetics at all, is that the same biological state that they're in, or can these we could have the two sets of people, two biomarker levels at the same region, but it's a very different biological state because the origins of that biomarker at that level are different.
Okay, so that's a great question because it brings up the fundamental issue of Mendelian randomization, which is somewhat complex to explain. But to me, the fundamental assumption of Mendelian randomization is gene-environment equivalence. And that is that the state that you're in will have the same downstream effect, whether it is influenced by a counterfactual genetic difference. I mean, obviously, we can't CRISPR you and then find out how your LDL cholesterol is. So it's a counterfactual difference, which is why, we compare you to your sibling who has different genetic makeup or whatever. That's the nearest we can get to the counterfactual. It would have the same downstream effect as if the factor was modified by any sort of intervention we'd like to do, like cutting down on the number of sausages you eat, or etc., or giving you a statin, or, a tablet which lowers cholesterol. Now, so the thing that I say journals should actually say is you've got to justify gene environment equivalence. In fact, there's publication guidelines on Mendelian randomization of what people should have to report to allow you to a reader to evaluate it. And one of the things this is the strobe MR, it's called. One of the things that they have to report is to justify gene environment equivalence. Now, in those cases, then that is perfectly reasonable to say that the effect of that difference in state on your coronary heart disease risk, it'll be the same effect if your LDL cholesterol is down 10% because of a counterfactual genetic difference, as it's down 10% because you've cut out the sausages. Mm-hmm. Then that is reasonable for LDL cholesterol. I mean, now there's other ways that you can get low LDL cholesterol. So, for example, developing cancers lowers your LDL cholesterol. But then, obviously, that would not have the equivalent effect, the sort of association with disease. And you can use the similar sort of arguments for body mass index. The genetic variant FTO influences calorie intake, and then therefore you sort of think that the genetic variant in FTO would be equivalent to a sort of intervention you would like to do to reduce body mass index, but you could reduce and that would be the case with calorie intake. You could reduce your body mass index, as Miguel Hanan comically pointed out, by chopping off a leg. Okay, that if you chopped off a leg, that would reduce your body mass index, but that ain't gonna be any good for your health. It's not going to give you a good outcome. So that's so those are examples where gene environment equivalence wouldn't apply. So it's justifying gene environment equivalence and understanding gene environment equivalence, which is absolutely key to a sensible or interpretable MR study.
Are there any sort of general or final thoughts you want to leave people with around, I guess, your concept of evidence triangulation, how they should think about, taking an integrated view of human health or just science in general by looking at different types of studies, each of which has different assumptions, different sort of standards of rigor and evidence and so forth. Which, in the context of thinking about human health, what do you want people to take away from this conversation, your work, in terms of what they need to do to be appropriately careful before drawing conclusions?
Yeah. So I think this goes back to what's been referred to as the variety of evidence thesis. And the variety of evidence thesis is that it's not the volume of data which matters. It's that you have different types of data from different types of studies, all of which can be biased. As I say, randomized trials can be biased. All studies can have biases. But if you just have a vast volume of studies that just repeat the same bias, then you will get the wrong answer. What's required is actually to think about how can studies have different biases. So let's think of randomized control trials of cholesterol-lowering drugs like a statin, and those randomized trials could be, they could be biased. It could be that when you give the treatment, that influences something else, which is in some contexts, which actually has the effect on the outcome. There could be biases in randomized trials, but then you have a Mendelian randomization study when it looks at genetic variants in the HMG co-reductase gene that relate to cholesterol level. and predicts the difference in coronary heart disease risk. The Mendelian randomization study can be biased by genetic pleiotropy, which won't influence the randomized control trial. And a study which uses a negative control to actually sanity check whether it's whether the observational association is at least has some face validity in terms of being causal. If your negative controls don't show up biased, that would make you then weigh those studies more heavily. Other approaches are things like cross-context comparisons, where you deliberately go to situations where the contexts are different. So, for example, contexts can be different countries, different places where confounding is different. I mean, in studies in breastfeeding, we looked at in Bristol, Avon longitudinal study of parents and children, breastfeeding in 1991. It was the mothers with higher education, families with better, in more favourable socioeconomic circumstances, different patterns of mistake of health damaging behaviours, etc. Breastfeeding predicted everything. Everything good was predicted by breastfeeding. But if you went to southern Brazil at that time, that wasn't the case. It wasn't, breastfeeding, still was not confounded in the same way. So you did studies between those two populations, and the things they agreed on were there. That you therefore had greater faith in the things they agreed on. Yeah. So it's just always thinking of, is there a variety of evidence in this whole in the studies that have been done of this issue? Is there a variety of evidence? If there's not, then you think we need varieties of evidence to address this question.
Yeah, and I think what you just said is important too for sort of another or related reason, which is virtually everything in biology is context dependent. Our biology adapts different contexts. That's sort of what the essence of it is. And very often when people are interested in medicine or diet or human health, some aspect of human health, they're often looking for something that is context invariant. They want a drug that will work for everyone, or in this part of the world, or that part of the world. So that's just another reason why it's important to think about, whether a result holds across different contexts.
Yeah, absolutely. And those are sort of situations actually where the genetics can be really helpful. I mean, so say you can't, statins have been trialed in more people than anything except vaccines in individual person randomized trials, I think. But even so, they haven't been tried in every country, every population group, etc. But you can sort of get genetic data in the different population groups, and if you see that the genetic variants in HMG CoA reductase that relate to LDL cholesterol level relate in the same way to coronary heart disease and without adverse consequences seen in these different contexts, you can then transport the findings that you get from the randomized trials to other places.
All right. Well, this has been a fascinating discussion. I want to thank you for joining me across the pond, George. Anything else you want to say, or any place you want to point people to if they're interested in this stuff or your work?
Yeah, so there's a recent paper in the European Journal of Epidemiology with Serena Gutierrez as the first author, and Maria Glymore and me on triangulation. That's recent. So that's probably the most recent sort of review of this of triangulation, and within that are then references to all.
Yeah, and I definitely I looked at that to inform this conversation. So I'll link people to that in the episode description. But once again,
and then you could, if people want to, they could read. I wrote a paper called the Mendel's Laws paper. I wrote it was also in the European Journal of Epidemiology. It's about the whole alcohol and folate and nutritional epidemiology sort of debacle. Maybe you could link those two in.
All right, great. Well, Professor George Davey Smith, thank you very much for your time.
Great. Take care.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.